We're going to move on to our next speaker, which is, which is Mieke. She's in, I think, probably the same lab and a co-author of Kristen, so she'll continue on with the theme of DNA metabarcoding and tell us a little more about it.
Thank you. Let me just get, so thanks, Kristen, for giving an excellent introduction. And I'll just be following up on Kristen's talk on what we did for the past couple of years, using DNA metabarcoding to look at invertebrates and their associated plant communities, and how we can use that to look at restoration trajectories in WA. So Kristen's already covered why we do monitoring. It's to assess restoration success, improve future restoration. You can do adaptive management and put interventions on if you know how successful your restoration is or unsuccessful. And also because there's a financial obligation to demonstrate that your restoration is successful before the tenements can be released. We've already talked about the field of dreams and the vegetation bias and a lot of restoration monitoring, because it is expensive, time consuming, and requires a lot of very specialized taxonomic expertise. This is especially true with invertebrates. So they're already very well-established indicators of restoration. People use especially like ants and such to as indicators of restoration because they're numerous, they're pretty easy to trap. And they're indicators of ecosystem functions like the detritivores or soil formation, pollination, herbivory, etc. However, they can be pretty expensive to identify because often it's a per specimen cost, and there's a lack of taxonomic expertise to identify all these invertebrates in the specimens. And the additional problem is that there's a lot of undescribed diversity. In Australia, approximately 75% of the arthropod diversity is undescribed as yet. So metabarcoding is one of the tools we can apply to invertebrate monitoring that could help. Sequencing technology is becoming cheaper and more available. And metabarcoding allows us to simultaneously sequence hundreds of species in the same sample. So that can be very helpful, especially because it's a per sample cost, not a per specimen cost. And this is because when you take your sample, say all the ants and all the insects in a pitfall trap, you blend that all together and you extract the DNA from that blended sample. So instead of trying to identify all the specimens in a sample, you're just sequencing the sample as a whole. Kristen has already gone over the metabarcoding workflow, so I'll skip that a bit. The limitations that are especially relevant for invertebrates are the unreliable abundance estimates. So you have to be sure that if you're going to try use invertebrate monitoring to look at restoration trajectories, that you can still do that using just presence-absence data, or using the number of traps a certain species was detected in as a proxy for abundance. And the other thing is the underdeveloped reference databases. So because the invertebrate community is so diverse, there's a lot of species that aren't in the reference databases. So those you can only identify to family or order level based on the other species that are in the reference database. And this can be a major limitation. However, you can still use those sequences and where they show up as your community data to see how the communities are changing over time. And it can still be a very valuable tool in biological auditing. So in Kristen's paper, we were able to see clear changes in the invertebrate community. However, we only used a single reference site per mine, and it was only in the Swan Coastal Plain, and we only used the pitfall traps. So the project that I'm talking about now, we're basically sort of expanding on that and seeing how it can be more relevant to places other than sand mines in the Swan Coastal Plain. So what we're looking at for trajectory was basically looking at differences. So we want to establish that there's differences between the restoration and the reference communities, and that there's direction, that the restoration communities are becoming more similar to the reference communities over time. That's what we would call recovery trajectory. So overall, we thought that the ground-dwelling invertebrates would show those recovery trajectories better than the airborne invertebrates, just because the airborne invertebrates have higher dispersal, so they would recolonize quicker, whereas the ground-dwelling invertebrates would show the local conditions better and not be coming from outside a restoration site from the nearby reference sites. The other thing is that invertebrate communities would show those recovery trajectories better than the associated plant communities. Because again, we're looking at the plant communities we get from the sequences on the invertebrates. So not necessarily the plants that are at that site, they could have been brought in from outside. And the other thing is that these trajectories may not necessarily be uniform across different ecosystems. We've shown that you can see those changes very clearly in the Swan Coastal Plain, but that may not be the case for somewhere very different, like in the Pilbara. So we went to 3 different locations, the Pilbara, the Swan Coastal Plain, and the Jarrah Forest, and we collected from chronosequences of restoration at each of those locations. So in the Pilbara, it has a very hot, arid climate with stony loams and low fertility. The reference sites have snappy gum over hummock grasses. In the restoration sites, you have a few more acacia bushes, but you generally also have those grasses popping up. The restoration was done by land forming using stockpiled topsoil on the top and ripping. In the Swan Coastal Plain, it's a Mediterranean climate. It has low nutrient sandy soils, and they're trying to restore back to the Banksia woodland. The restoration here was land forming, a direct transfer of fresh topsoil, ripping, and seeding of native species. And this is one of the sites that Kristen also used in her invertebrate study. Finally, in the Jarrah Forest, it is also a Mediterranean climate, and it has those lateritic soils. The restoration, similar to the Swan Coastal Plain, was land forming, stockpiled and fresh topsoil, ripping, seeding, and transplants. So for all of these, you'll notice that we have 2 reference communities, and they're spatially separated. So generally, I try to have one of those reference communities be north and the other one south of all the restoration sites. And this is because in our previous study, we noticed that although there was an increase in similarity with the reference site with time, so the older sites are more similar to the reference site, the older sites were also closer to the reference site. So we had that spatial autocorrelation problem. So in this project, we want to separate the effect of distance and community similarity to be sure that it's the age of the site that is causing that increase in similarity and not the distance to the reference site. So for sample collection, we collected from 2 different types of traps: vein traps to collect flying insects and pollinators, and pitfall traps. There were 2 vein traps per sample and 4 pitfall traps per sample and 5 samples for each site. So that's the restoration sites and the reference sites. So in total, there were 200 samples because the vein traps and pitfall traps were kept separate. And we used an invertebrate assay, an improvement on the previous one that also gets Hymenoptera, and also a plant assay to look at the plants that were associated with the invertebrates there. So in total, we got 14 million invertebrate sequences that after filtering gave us 951 ASVs. So ASVs are basically our sequencing species unit. It's called an amplicon sequence variant. And you can have multiple ASVs per species, just from natural genetic variation. So it gives a little bit more detail than just species. But the reason I do ASVs in this one is because a lot of them I couldn't get the taxonomy further than order or family level, again, because of the reference databases. So for this project, I'm looking at if we can establish those trajectories with the limited taxonomic information that we get, using mainly ASVs. For the plant sequences, we've got again about 13 million plant sequences and 205 ASVs after filtering. For both of these, you can notice that the pitfall and the vein traps can be pretty different, and there's also very clear differences between the different locations, between the Swan Coastal Plain, the Jarrah Forest, and the Pilbara. So first, to establish that there were differences between the restoration and the reference sites, we did an ordination with permanovas. So it's NMDS ordination, and the lighter colors show the younger restoration, the darker colors show the reference sites. And ideally what you would see is the reference sites clustering together and the older sites being closer to the reference sites than the younger ones. You can see that for some of them more than others. As we predicted, you have tighter clustering in the pitfall traps and more overlap in the vein traps. Again, this is because the vein traps can capture invertebrates from a much greater distance than the pitfall traps because they have higher dispersal. And the plant communities has a similar pattern of there's a lot more overlap than in the invertebrate community because the plants can be brought in from outside as well. I just wanted to show you this one as well. I divided the ASVs into the ones that were present in the reference sites and the ones that were not. So the blue is the ones that showed up in the reference sites and the red is the ones that weren't. And this is just to show you that the differences between the sites is driven mainly by compositional changes rather than any changes in richness and such. So finally, to look at trajectory, we're looking at the similarity between the sites and the reference sites, and they're based on their ages. So for a recovery trajectory, we want to see that the older sites are more similar to the reference sites than the younger ones. And this was true for the pitfall traps of the Swan Coastal Plain and the Jarrah Forest, but not in the Pilbara. It was also true of the vein traps in the Swan Coastal Plain, but there's a weaker relationship there than in the pitfall traps. So overall, the pitfall traps do show your recovery trajectories better because they reflect the local environment better, and they're less likely to be drawing in flying insects from outside your restoration area. It's good news because it is showing that the invertebrate communities can recover without intervention, although this is dependent on there being source populations. All of these restoration sites were adjacent to native vegetation, so that could have helped with the recolonization. For the plants, we still saw some of those recovery trajectories in some of our locations. And it is an indication, along with the ordination, that the invertebrates are likely foraging locally in the restoration sites, which is excellent news. However, in the Pilbara, we don't see any of those types of relationships. And part of that is the Pilbara is already a pretty harsh environment. It's hot, arid, and it has a lot of open space already. So the invertebrates there are likely already adapted to those open environments, and it's easier for them to recolonize rather than in the Jarrah Forest or the Swan Coastal Plain, where you often need an accumulation of things like litter for those detritivores and such. And finally, the thing that I wanted to do in this project was to separate the effect of age and distance, which is the reason we had those multiple and spatially separated reference sites. So we looked at the variance that could be explained by either age, restoration age, or the distance to the reference sites. And you can see in all the locations where we could see a clear relationship between the age of restoration and the similarity to reference site, the restoration made up a lot more of the variance than distance. And it's only distance was only a factor in those locations where there wasn't a relationship between age and similarity to reference. There are some limitations with this project as well. We only had one chronosequence for each of the locations. So that's a limit of replication, but it can still be it's a case study of what you might expect in these different climates and ecosystems. The reference databases, I think, will always be somewhat of a limitation, and it's only by building up those reference databases that we can expand on the data that we can get from these kinds of studies. The other thing is to look at seasonal variation. So I only did this once in the early summer, springtime. So it's kind of the ideal time to look at invertebrates and such. But Kristen's one was done in March, so it's very different. She also did a plant assay, but didn't find the same sorts of patterns that I did. So for there, it could be seasonal variation causing differences in the results that you get. The other thing is that we use chronosequences rather than long-term studies. So if you wanted to look at how communities change over time, it might be better to look also at some long-term studies to see how relevant they are and not based just on sites. However, we can still say that pitfall traps can show recovery trajectories and more clearly than vein traps. However, the patterns of invertebrate recovery are not consistent across locations. This may not be as helpful in the Pilbara compared to in the Jarrah Forest or the Swan Coastal Plain. Overall, invertebrate metabarcoding can reveal restoration trajectories, but there are caveats, and we need to know those caveats in order to make the correct inferences from our data. So thank you very much, everybody who helped me, CMSR and all the people at the mine sites that helped me facilitate my field trips.
I think we have time for one or two quick questions, if anyone has any.
Hello, thanks for that. One quick, well, I guess I have two questions if nobody else does. But the first one is, when you were talking about the ASVs, you were saying that these, like this one species can have more than one. So I was wondering, are all the samples at that level considered the same sort of like taxonomic level, per, like, to say it some way? Or is it mixing and matching, you know, like, by analyzing everything at ASV level, are you analyzing the same taxonomic, like, I know it's not a taxonomic level by itself, but you know what I mean?
What you're talking about, like if I'm analyzing species and orders together, or I'm analyzing at one level. Yeah, so the ASV is analyzing at the ASV level is one way to make sure it's at mostly the same taxonomic level. It includes some variation because there can be a couple extra ones per species, more in one than the other, but at the same time it is more of an equal taxonomic level than trying to get your taxonomic identification and then concatenating it that way, because for some I could only get to family or order level. So if I put all of those ASVs together, that would kind of be bulking up that order, whereas the ones that I got to species level would be,
yeah, exactly. That's
So that's why I kept it at the ASV level, to make sure it was as even as possible.
Okay, thank you.
I think that's all the time we have for questions. So thank you, Mieke.
Thank you.